AgTech 360 discusses breakthrough technologies that are impacting growers, businesses, and consumers. Hear from industry and academic experts about what's on the horizon.
So welcome back to AgTech 360 and our Biologicals in Action series. Today we're exploring how RNA technology is opening a new chapter in sustainable crop protection. Joining me is Mark Singleton, CCO of GreenLight Biosciences, to explain how RNA interference, or commonly known as RNAi, works, why it's such a promising alternative to conventional chemistry, and how GreenLight is using this breakthrough to develop precise biodegradable tools for growers. Thanks for joining me, Mark.
Nice to be here, Adrian.
So for listeners who might not be familiar, can you explain in simple terms what RNAi is and exactly how it works?
You know, if you dig down into the details, it's a very complicated process, but in essence, RNAi is a naturally occurring mechanism that happens in almost all living organisms, and it's a way by which cells regulate gene expression. The way it works, so the genes are contained in the DNA that sits inside the nucleus, and then there's the RNA, which copies the gene that's in the DNA, brings that, and then transcribes it, helps it be transcribed and turned into the requisite protein that gene codes for. And so if you think about, you can think about DNA and RNA in the same four Lego bricks. So there's one color Lego brick, four different color Lego bricks, and they're all stacked up, and a gene is many, many, many of these different Lego bricks, each Lego brick representing a nucleotide. And so what happens is, in RNAi, the RNA piece, the bit that's done the transcribing of the gene, is chopped up by naturally occurring processes in the cells. And where those sequences, when they reach about 21 Lego bricks long, 21 nucleotides long, they correspond with RNA that is currently being transcribed from the DNA. It says, hang on a minute, we've already made this protein. We're going to stop making the protein. We're going to shut this gene down. Okay, so that's the way I think about it in very simple terms. And so what that means is we can introduce from another source an RNA that is a mirror image, if you like, of that gene. That RNA gets taken to cells, it gets processed, it gets chopped up, and where you get these corresponding sequences, the cell believes it's made that protein, and so we silence that protein. So in essence, unlike many conventional crop protection chemicals, which interfere with the protein after it's been made, what we do is stop that protein or enzyme being made in the first place.
Before we get to all of that, tell us a little bit about your journey. I mean, how did you get interested in this type of biology? I know a bit of your background, so I know you've done a lot of things in the crop protection area and business.
I was working for a company that doesn't exist anymore, which was Uniwell Chemical, or probably CK Whitco at the time, became Chemtura and eventually acquired by Platform and then UPL. But I had the job title of the new product research manager. And I can remember reading some papers about this, and I was a little bit behind the times, but this new concept called RNAi, which had been discovered by Craig and Mello and Fire a few years before. And I can remember reading some papers and thinking, that would be really cool tech. And then reading a little bit more and thinking, yeah, you're going to need some pretty deep pockets to explore this. So maybe my little job here at Chemtura, a small company, about a $400 million business, was not the place. I just knew I wasn't in the right company. My interests were making either existing products more efficient, more effective, or finding biologicals that were ever more efficient and more effective. And this seemed to fit that career thread that I was following for the longest time. And so I'd explored avenues to get into RNA, but I was never successful in getting on board. So now we'll fast forward to Arista, and eventually this company called GreenLight comes to me, and it was just around the time Arista was being sold to UPL. And they say, hey, we've got this new way of making RNA. It's called a cell-free reaction. And we think we can make RNA at less than $1 a gram, and we think that makes it competitive. Are you interested? This time I'm in a situation where I can say, yeah, I think I'm definitely interested in this. I would like to learn more about how you intend to turn this into products. Anyway, fast forward those conversations, a number of different things happened in the industry, and eventually one day, I was asked to join GreenLight. So that's how I ended up here. It's kind of been almost a career-long journey.
So that brings me on to my next question, which is a kind of big picture question before we maybe delve down a little bit more into what GreenLight's doing. But, how should RNAi be leveraged in agriculture? And, are, what makes it such a exciting technology from your perspective?
I always tell people that RNA just doesn't want to be a pesticide. Okay, it just doesn't want to be a pesticide. It just wants to be left alone and go copy and paste, that gene transcription producing proteins. And so you have to convince it to be a pesticide, to have the effects that you want. And there's a number of reasons for that. One of which is, I used to say that, we were all here just to replicate DNA, and having read the selfish gene theory as a younger scientist, I now come to the conclusion we might actually be all here to replicate RNA. So we're all here. We are all RNA recycling machines, as is pretty much every organism on the planet. So everything that exists that is alive knows how to handle RNA. And that's a real boon from a pesticide perspective, because now you know exactly what it's going to do when it comes in contact with other organisms. So it makes it very exciting. And the fact that you can be very highly specific makes it even better. So you are able to minimize any off-target interactions.
What is GreenLight doing to make sure that consumers are comfortable with these types of biological technologies, which, as you've described, have a lot of advantages over some of the chemical alternatives, but they are new and they are different?
The first strategy really is to keep it simple. So wherever we can rely on what we term naked RNA. So we don't embellish it with things, we don't change any of the molecule. The RNA is exactly the same as RNA that you could stumble across in the environment. There's no molecular changes, no atoms changes. It is just the RNA. Having high quality products, so we have a specification, we know what we make, we state what we make. These are all, to me, the very fundamentals that we have to do internally if we're going to be credible. The next thing we have to do is to have good data. And that's very difficult for a startup company that is pre-revenue or just on that. We have to have a lot of data that shows that we know exactly what happens when we release our product into the environment. That's what a spray application event is. It's an environmental release. We need to be able to clearly show this is what happens. And so we try to do that. We do a lot of outreach. We have a lot of presentations to the scientific community, the regulatory communities, but also food, various different food environments, formats, venues, conferences. We'll reach out and talk and explain.
The North Carolina Plant Sciences Initiative impacts lives through innovative applications and discoveries. By leveraging cutting-edge research and technology, we address global challenges related to agriculture, sustainability, and human health.
So let's talk about the regulatory landscape. And I think it's fair to say that for biologicals of various types, it is not as evolved in many cases. How do they compare with chemical alternatives? Are they difficult, or what has that been like?
So with RNA, the one thing that is a little bit different is agencies want to see more of the data that's classically associated with the AI, also with the AI and formulation. And that's because, as I said, the formulation is the key to unlocking RNA. So they're very interested in that piece. So that's a little bit different. We found that when we're able to show a dossier and submit a dossier, the agencies are very keen to speak with us and to understand, or to take our understanding of RNA. And so we've had very deep dialogue with regulatory agencies across Europe, obviously, first and foremost in the US, across Europe, in Mexico. So lots of interest from regulators in trying to understand what is important when they're trying to figure out the risk-benefit analysis with RNA. The first one was a long journey. The second one, with a much more refined dossier based off that feedback, was a much smoother journey. We did a much better job of doing our job right and doing it right first time. So I think we're now starting to understand, at least in the US, exactly what people want to see to measure that risk benefit. It's a little bit different in all the other jurisdictions. We're trying to make sure that we're able to meet that.
So, looking forward, and maybe let's take an arbitrary period, say the next decade. I mean, where will RNAi technologies be at that point in time? Are you seeing them, replacing synthetic chemistry, complementing? How do you see them play against other biological-based products? What's it going to look like?
So that's a great question. It really is. So when I tend to speak about RNA, I speak about my job is to offer growers a viable choice, right? So that they have reliable control and they have a light, deft touch in the environment. So they get that viable choice. If you look at the RNA form and what we're currently doing, then I estimate that with the technologies as they currently stand, I can probably access about 2/3 to 3/4 of the entire crop protection market with the RNA products using the technologies we've got. There are some places that I can't go yet. I think in 10 years' time, the ability for RNA to get into those spaces, either through the formulation biology that we've discussed or alternative different designs, structural designs of the RNA will see us complete that journey. I also think that the ability to affect output traits on crops in the same way that biostimulants do, but with a much more targeted approach. Do we want to mitigate a period of drought? Do we want to mitigate a period of heat stress? Do we want to increase dry matter content? Do we want to build carbon? Do we want to build protein? Do we want to add sugars? These will be things that we can also reach with RNA, as long as we're careful to maintain and bring with us those people who give us that social license. So I think RNA will be a big piece. I think if I've done my job properly, we'll have a lot of RNA competitors out there all looking to maintain that social license, all looking to maintain that performance. And I think the ability that GreenLight has had to get these products through the regulatory agencies and get them adopted, hopefully that's inspiring some other companies to bring forward a new generation of biologicals. And I could start rattling them off, but they know who they are. They know who I'm talking about. There's some really cool companies out there, and having them come forward and bring forward that viable choice, I think that helps reduce the load in the environment that we need from conventional crop protection products, and replace them with more, as I say, things that have a softer environmental touch, because farmers have that choice. And so I'm very excited that we'll begin that program to balance out. I don't speak about wholesale replacement because I'm a bit of a pragmatist, but I think offering that choice, and when people see that viable choice and that balance, then we'll see a transition that enables biological materials to start to replace a lot of what effectively is a very essential need that we have if we're going to secure the food chain, and that reliance on conventional chemistry. I think we'll start to see that dependence and that reliance shift. Will it eradicate 10 years? I don't think so. The regulatory process is that long in some places for some of these things, but I think we will be much more optimistic about being on that journey.
Mark, thank you.
AgTech 360 shares relevant news and breakthroughs with audiences across the globe. Stay connected and join the conversation by following NCPSI on social media.